はい、それでは定刻になりましたので、ウェビナーの方を始めさせていただきたいと思います。バイオストリームのシングルセル解析ウェビナーを始めさせていただきます。本日大変お忙しい中ですね、弊社のウェビナーにご参加いただきまして誠にありがとうございます。本日司会を行いますアプリケーション担当細野と申します。どうぞよろしくお願いいたします。はい、で、本日のウェビナーでございますけれども、ピクセルジェン社というですね、スウェーデンの会社のですね、シングルセルのプロテオミクスに関連するキットのご紹介になっておりまして、ちょっと今、スライドがちょっとフリーズしてますね。すみません。フリーズして。行きましたね。はい。で、こちらのですね、ピクセルジェンのプロキシオムキット、イムの155というですね、キットについてのご紹介ということになります。はい。で、こちらのキット簡単にご紹介させていただきますとですね、NGS ベース、シーケンスベースのこれキットでございまして、シーケンスでタンパク質を検出するというキットになっています。で、特別な装置を必要としないという特徴がございまして、ラボに常設されている機器で実験をしていただくことができるというキットになります。はい。で、標的タンパク質はもう155種類ということで、非常にたくさんのですね、タンパク質、細胞表面のタンパク質になりますけれども、それを解析することができまして、細胞数としましては、この1000細胞をですね、解析することができるというキットになっています。はい。で、サンプル自体はですね、細胞サスペンションになりますので、サスペンションにしていただいてサンプルを使っていただくということになります。はい。で、最大の特徴はですね、こちらにございますような3D マップですね。これは分子レベルでの 3D マップをですね、描くことができるというのが最大の特徴になりまして、細胞あたり5万分子のですね、そのタンパク質の相対位置を特定することができるというキットになっています。はい。で、相対位置が分かると、何が分かるのかということなんですけれども、通常のタンパク質のですね、発現量だけではなくて、こちらにございますようなですね、その細胞表面タンパク質に関しまして、その細胞表面上で分散してですね、発現をしているのか、それともクラスター化をしてですね、発現をしているのか、またはその極性化してですね、発現をしているのかと。で、そのタンパク質の細胞表面上での状態を知ることができるというのが一つ特徴としてございます。で、またもう155種類というですね、非常にたくさんのタンパク質を解析をしますので、そのどのタンパク質同士がですね、この共局在をして、分子間相互作用しているかというところまで知ることができるアッセイということになります。で、近年、免疫学の分野ではですね、多くの細胞表面タンパク質が自己クラスター化をしてですね、で、免疫反応を活性化するということが報告されています。で、例えば、こちらのヘルパー T 細胞の系では、抗原認識の際にですね、この TCR が自己クラスター化をしまして、で、免疫シナプスというですね、構造を形成して活性化することが知られておりますし、CAR-T などでもですね、CAR-T、カーマイクロクラスターというですね、構造を形成しまして、がん細胞を効率的に攻撃するといったことなどが報告されています。で、こういった形で、このクラスター化というのがですね、この様々な細胞間の相互作用に関わっているということが報告されているということになります。で、この極性に関しましてはですね、これは以前から報告されていることでございますけれども、細胞表面では複数のタンパク質が共益をしましてですね、共益分子としてこの機能単位を構成をしまして、で、細胞間相互作用がコントロールされているということが知られています。はい。ですので、このようにですね、細胞の状態とか機能を正確にですね、把握するためには、単に個々のタンパク質の発現量の変化だけではなく、この細胞表面のですね、この空間的、動的な変化をですね、幅広く捉えられるようなアッセイが必要ということが、最近言われるようになってきているということになります。で、本セミナーでは、これをですね、実現できる PNA アッセイ、プロキシオム、ネットワークアッセイ、近接ネットワークアッセイとも呼ばれておりますけれども、これについて詳しくお話をさせていただきます。はい、それでは早速メインセッションに移りたいと思います。で、ご質問に関しましてはですね、チャット機能がZoom にございますので、そちらからお願いできればと存じます。はい、で、ご質問は日本語でも大丈夫です。で、セミナーの最後にQ&A セッションで回答をさせていただきます。はい、で、そうしましたら、ここからはですね、英語の方にちょっと切り替えさせていただきます。OK, let's get started. Now we move on to the main seminar by Dr. Hannah Van Ooyen from Pixelgen Company. And the seminar title is a Single-Cell Protein Interactomes by Proximity Network Assay. It's my great pleasure to introduce Dr. Van Ooyen. She's an immunology application scientist at Pixelgen. She obtained a PhD from KTH Royal Institute of Technology, and she is expert in immunology and single-cell assay. Yeah, Hannah, thank you so much for joining us today. I hand over to you.
Thank you very much for that kind introduction. Okay, so let's get into it. The plan for today is to introduce you a little bit to the technology that we have developed at Pixelgen Technologies. So I will guide you through both the background as to kind of why this technology has been developed and what areas we're filling in the field of proteomics. And then I will show you how the technology works and also showcase some application examples so you can really see how the data can look like, what type of biological questions you can address with this technology. So, as mentioned in this nice introduction, this is a method for protein interactomics, and it's called the proximity network assay. And this was developed by Pixelgen Technologies, the Swedish biotech company. So why do we want to look at protein interactomics at all? First of all, we're very much interested in proteins. As immunologists, we know that protein is really the molecule that governs cellular function, that controls cell responses, and membrane proteins play obviously a very vital role in this part, being kind of on the verge of the cell limit, communicating from the inside to the outside of the cell. But typically, it's not really enough for the cell to just have a protein. That's not going to make this protein signal. But instead, for a protein to really carry out its function, it needs to bind ligands, it needs to form multi-protein complexes, it needs to cluster, it needs to reorganize, and this is how signaling is induced. And this is particularly important for immune cells, since immune cells require very directed responses. It's, for example, with cytotoxic immune cells, it's of course very important that they target the diseased cells and they don't damage any healthy tissue in the surroundings. So these responses need to be very directed. And the way that the cell achieves this is through spatial reorganization of membrane proteins. And to study this, we really need to start looking at the field of protein interactomics. So this allows us to look at how proteins are organized and to really capture the complete picture of cell function. So traditionally, if you want to look at protein organization, the methods kind of to your disposal have been different microscopy-based techniques. So with recent development in super resolution microscopy, it's possible to have very high resolution to really resolve structures. But typically, you need to know what you're looking for. It's very hard to do discovery studies since you can only stain a few proteins at the same time. This really requires you to know kind of what you're searching for. On the other hand, if you want to do more discovery-based studies and do high multiplexing, you can use different high-dimensional flow, mass cytometry, or sequencing-based proteomics methods, which will give you information about a lot of proteins. But on the other hand, you completely lose the information about how these proteins are organized. So being back to a traditional proteomics approach. So this is really the gap that we wanted to bridge at Pixelgen Technologies. And this assay, the proximity network assay, it is really doing this by both providing very high multiplexing, so you can look at 155 proteins at the same time, but then also providing spatial information for all of these proteins actually at a very high resolution. So in a typical PBMC cell, you will get around 40 to 50,000 unique molecular zones, so to say. I will show a little bit how the data looks like. What is really great as well is that you have very high throughput. So for all of these markers, 155 markers, you will get information in all of your cells at the same time. So there is no need to kind of scan through, look at individual cells one by one as you would typically do in microscopy, but rather you would get this information for all the cells. And in order to really handle this quite complex data, we have developed scores that assess protein organization. So we measure clustering and we measure colocalization as well as abundance. And this analysis can be performed using a free software. So we have developed a free software that is open source. It's open to use for anyone, basically. So what we provide is a sample prep kit. It's not an instrumentation. So what you do is that you go from a single cell suspension, you apply this kit, these chemical reactions, and then you sequence these libraries. So there's no microscopy involved at all. It's completely sequencing based. And then through data processing and analysis, you can regenerate the spatial map of every single cell in your sample. So here, for example, you have a single immune cell or even a cell complex where you have two cells interacting with each other that we have visualized with this assay. So how can we generate these three-dimensional kind of protein maps on the cells? How do we do that by sequencing? So the first step is to put your cells in suspension. This is a method for cells in suspension, so not tissues. And as a second step, we fix the cells. And the reason why we do this is really to immobilize the cell surface proteins so that they cannot move during the rest of the protocol. After this, we add our 155-plex antibody panel. And in this panel, every single antibody has a small DNA sequence conjugated to it. And this sequence is unique. So every antibody has a completely unique sequence, making it possible to identify then individual proteins. In the next step, we do some rolling circle amplification of these small DNA sequences, and that is done in order to make them a little bit longer. And once they are a little bit longer, it enables us to actually connect sequences that are very close to each other in space. So for two proteins that were close to each other, we will be able to connect their two sequences. And then this is done over the entire cell surface at once. So you can imagine that we're forming these very complex networks linking all the individual proteins to each other. And then what we do is that we sequence these linkers. So basically every linker here contains information about which two proteins it has linked. And through data processing, we can then informatically rebuild the cell surface of every single cell. So if you input 1,000 cells into this assay, you will build these networks for 1,000 cells, and you will computationally then identify 1,000 individual cells in the data analysis process. So regarding this panel, the 155-plex panel that we're using, this is a very broad panel, so it's aimed at enabling analysis of heterogeneous immune cell populations. So it's very suitable for example, PBMC samples, bone marrow samples. You will detect major cell populations in these T cells, B cells, NK cells, monocytes, DCs. You can even use it for stem cell research or look at other immune cells like granulocytes, platelets. We have also quite some experience with different cell lines. So it's very versatile in the type of cells that you can analyze. We've also tried to select targets in different areas. So we're targeting both adhesion molecules, cytokine and chemokine receptors, different activating inhibitory receptors, proteins that are important for regulating the organization of other proteins, etc. So it's a really broad panel. You also do have controls in terms of kind of protein complexes in the assay. So of course we have isotype controls, but we also have proteins that target two different subunits of the same protein. So you can use this for kind of validating the co-localization scores. I will show you this in a second. You can see how it looks. So when it comes to the actual output that you get from one of these assays, this is a protein-based analysis, and it's also single-cell-based analysis. So similar to other single-cell proteomics methods, you will get protein abundance for each cell. But then what is really novel is that you will also get then this information about protein organization. So both looking at protein clustering for each protein and also looking at pairwise co-localization. So for the protein abundance, this can be used to, for example, detect different cell types in your sample. This is a PBMC sample that we processed with the method. You can see that we can really detect different key cell types in PBMC, like naive, memory, T cells, Tregs, different dendritic cells, even some contamination of different granulocytes. You can then, of course, for every single cell in your sample, visualize this cell in 3D. So here you have an example of a naive CD4 T cell, and then we can really look at the distribution of any marker of interest. So for example, here we're showing you CD45, which has a very nice even distribution on this particular cell. You can compare your different cell populations in terms of protein expression profiles, looking at all of these 155 markers. Of course, you can also look at what happens to protein abundance in the cells when you stimulate or when you treat with a drug, or comparing samples in other ways. We've also done cross-validation against maybe the gold standard of single-cell protein analysis flow cytometry. So here we assayed some resting T cells and compared them to activated T cells. And then we looked at the expression of CD25. So CD25 is an activation marker that is upregulated during activation. And you can see here that we detect this upregulation very nicely in the activated cells. And the results are very similar when we did this analysis with flow cytometry. So you see that the protein abundance data is very, it's very similar to flow cytometry data. When it comes to the first spatial aspect, so protein clustering, this is basically measuring for a single protein how it's distributed over the cell surface. So we have developed a score to assess this. So if a protein is evenly distributed on the cell surface, you will have a close to 0 score. And if the protein is clustering on the cell surface, you will get positive scores. So one of the first applications where we kind of tested this assay was looking at the mode of action of pharmaceutical antibodies. So rituximab is a drug that targets CD20 on B cells, both in autoimmune settings and in cancer settings. And it's very much known that when rituximab binds CD20, it actually clusters CD20, and this is part of its mode of action, so it's very important for its function. So we treated cells with rituximab and we assayed them with our system to see if we could detect this clustering. And indeed we could. So here I'm showing you the protein clustering score for individual cells, either in the stimulated sample or in the control sample, just stimulated with an isotype antibody. And you can really see that CD20 clusters only in response to rituximab. And on the other hand, if you look at a control target like HLA-DR, we don't really see any induction of clustering. We also validated this with microscopy. So here in red, you can see that in the control cells, CD20 is very evenly distributed on the cell surface, whereas after stimulation, you see this very profound clustering of the protein. For the second spatial analysis, which is protein co-localization, we look at 2 proteins and we assess, are these 2 proteins organized in relationship to each other in a random fashion? Then we will have a close to 0 score. If they're actually co-localizing more than you expect by random chance, you will have positive co-localization scores. And on the other hand, if the 2 proteins are segregating from each other, you will get negative scores. So since this is done in a pairwise fashion, this allows you to look at the formation of protein complexes. For example, you can see the formation of different integrins containing 2 subunits. You can see interaction partners like the T cell receptor interacting with CD3, for example. But what is really, really powerful is that since you do this pairwise comparison between all the 155 proteins, it allows you to also look at much more complex structures. So really looking at multi-protein domains and how many different proteins are organized in relationship to each other. So really providing you the complete picture of the cell interactome. So this is an example of how you can visualize the data. What I'm showing you here is a co-localization heat map where every square here represents a co-localization score between 2 proteins. So we can zoom into a small selection of proteins here. So for example, here we have the co-localization score between HLA-ABC and beta-2M. And you can see that it's red, meaning it has a positive score. So these two proteins are co-localizing. And this is very much what we expect, since we know that HLA and beta-2M are part of the same complex. You can see other control complexes, as I mentioned, like LFA1 consisting of CD11A and CD18. We also have positive co-localization. When you look at the complete co-localization heat map, you can start to see patterns of several proteins that all have positive co-localization with each other. And these are representing these multi-protein domains, so where multiple proteins are all present in the same domain on the cell surface. And then you can look at how these domains are changing over time with stimulation in different disease settings, etc. We have, of course, also validated this co-localization results with microscopy. So we performed some super resolution microscopy where we targeted CD55 and CD48 that we noticed in our data was positively co-localized. And then we included control protein CD7, which didn't really show any particular co-localization with either CD55 or 48. And when we visualized these T cells with super resolution microscopy, we could see that both CD55 and CD48 were forming these nanodomains or nanoclusters, and they were highly co-localized. On the other hand, CD7 was showing this very even distribution all over the cell surface, not really enriching to the same clusters as CD55 or 48, really agreeing with the data that we could capture with the proximity network assay. So we have recently published a preprint that describes this technology more in depth. It's also in review in Nature Methods. So here you can see some other examples, validation of the technology, and really read in depth about how it works. So feel free to look at this after this seminar. So now to the maybe most important question: how can you apply this method to really, or to use it in your research? So providing this new dimension, this spatial information about proteins, it really allows you to get kind of novel insights about your cells and your diseases. So, for example, you can do much deeper cell profiling and disease profiling, identify new cell subsets, new cellular states that are characteristic for your particular setting. It, of course, also allows you to unravel molecular mechanisms, so really understanding, for example, resistance development or mechanistic functions of particular proteins that you're interested in or of drugs. You can use this to study mode of action or to do drug target identification, for example, identifying bispecific targets or identifying clustering proteins that could be targeted for therapy. This new dimension also allows you to look at completely new types of biomarkers. So not only looking if a protein is expressed in your disease setting, but to actually look at maybe it is expressed to the same degree, but it has a different organization, and this can be used as a biomarker and as a patient stratification strategy. So we have customers in all fields of immunology, hematology, immuno-oncology, immunotherapy, autoimmune diseases. So basically anywhere where you're looking at immune cells, you can apply this method. So now I would like to go into a few application examples just to showcase a bit more in depth different projects that have applied this analysis. So the first example I would like to guide you through is within the field of immunotherapy, specifically looking at CAR T cells and how you can use this technology to really profile and understand the protein landscape of a CAR T cell product. So for CAR T cells, it's actually known that it's very important how the CAR receptor is organized. So, for example, if you have a clustering of CAR receptors, which you often do with different solid tumor targets, you can have induction of tonic signaling and an exhaustion. So understanding how the CAR receptor is organized and how this is changed depending on the engineering of the receptor can really provide information about how the cells will function in vivo. And understanding the complete picture, so how all of the receptors are organized in a particular CAR T-cell product, can make it possible to even further predict responses in specific patients, etc. So in this particular study, we were interested in looking at CD19 CAR T cells and apply this method to characterize the organization of the CD19 CAR. So as a first step, we made sure that we could identify the CAR T cells in our sample. So we had a sample that contained both PBMC cells and CAR T cells. And then we did some high-dimensional clustering based on the protein abundance. And you can see how we can nicely detect the different immune cell subsets of PBMC and also detect the CAR T cells. We then measured the organization of the CAR receptor on these CAR T cells. Here I'm showing you one example of a cell where you can see the CD19 CAR in green. So this receptor is very evenly distributed on the cell surface, which has also been reported before. So that is the expected pattern. We then looked at all of the proteins that were expressed in these CAR T cells. So it was about 64 proteins that we could see expression of. And here again, I'm showing you one of these co-localization heat maps. So it really provides you a broad picture about how all of the different proteins are organized. You see some multi-protein domains here. You can really identify which proteins are co-localizing with each other. We were particularly interested in the CAR receptor, so we focused on the CAR receptor and looked at, okay, which interaction partner does the CAR receptor have? So here you can see the proteins that showed positive co-localization with the CAR receptor. We identified different adhesion proteins like ICAM1, ICAM2, ICAM3, also different co-receptors, CD8, some receptors that are very important for regulating TCR function like CD6 and CD5. And interestingly enough, also co-localization with the native TCR. So it seems that the CAR receptor, the CD19 CAR receptor, is integrated into a similar domain as the native TCR. We also looked at what proteins are actually segregating from the CAR. So for example, we could see that the CAR was not really present in lipid rafts. So here we have 3 lipid raft-associated proteins. We didn't really see any co-localization. Also, we didn't see any co-localization with these tetraspanins, and these tetraspanins are actually known to be important for regulating TCR function, but they don't seem to play a role, at least in the resting state, with the CD19 CAR. As a next step of this project, we were interested in knowing what happens to the cells during stimulation. So we co-cultured these CD19 CAR T cells together with CD19 expressing tumor cells, Raji cells in this case. And then we analyzed the cells after either 4 hours of co-culture or after 24 hours of co-culture. So first we assessed the changes in protein abundance. So we discovered that already after 4 hours you could see quite some changes in the protein abundance profile. This was particularly prominent after 24 hours. So for example, we could see upregulation of different checkpoint proteins like TIM3, PDL1, PD1. We also saw quite a drastic downregulation of the CAR receptor itself. Quite interestingly, we also saw downregulation of both the TCR and the CD28. And in this setting, we don't really have any antigen specificity. So it's interesting that the TCR is affected anyway. And one reason for this could be that the fact that the TCR is located very close to the CAR, and the CAR is being engaged and being downregulated, possibly affecting then also the TCR receptor. Another protein that we saw big changes in protein abundance was ICAM1. So ICAM1 appeared to be highly upregulated in the co-cultured T cells. So we then went ahead and visualized some of these co-cultured CAR T cells. And what we noticed was that ICAM1 had this very patchy phenotype. So part of the protein was localized all over the cell surface, but then there was a very kind of dense region with these patches on the cell surface. And these patches were also rich in B-cell specific markers like CD40, CD21, CD22, whereas they were often absent of T-cell specific markers like CD8. So this really informed us that ICAM1 is in fact not really upregulated on the surface of the cells, but rather it's acquired through a process called trogocytosis. So when cells are interacting with each other, they can pull membrane from each other, and this can result in transfer of proteins from one cell to another. And this is a process that have been reported in CAR T cells and that really influence cellular function. So it can lead to exhaustion in the CAR T cells, and it could lead to antigen escape of the tumor cells. So the tumor cells are transferring all of their antigens, making them impossible to detect, basically. So we went ahead and we quantified the degree of trogocytosis within individual T cells. So we noticed that some cells were not really acquiring any tumor patches, some were acquiring 1 patch, 2 patch, 3 patches, etc. And then we also divided our T cells into 3 different subsets. So looking at either non-activated T cells that were CD25 PD-1 negative, looking at activated T cells that were positive for CD25 but negative for PD-1, and those that we called exhausted that were positive both for CD25 and PD-1. And what we noticed in this analysis was that it was really these exhausted PD-1 positive cells that were acquiring a lot of tumor patches, much more than the non-activated. So this is a way that you can also assess cellular function using this spatial information. What is really neat here is that since you can really visualize these patches, it allows you to distinguish between cells that have gone through a lot of trogocytosis as compared to real cell-cell complexes. That type of differentiation would be pretty much impossible if you just look at protein abundance. So we can also detect these protein complexes in the data. So here, for example, you have a T cell that is in a conjugate with the Raji cell. So you can really see this by looking at T cell specific markers like CD2 being completely distributed on the T cell, whereas you have these B cell specific markers like CD40 being distributed on the B cell. So this type of analysis allows you to look at which cells are forming complexes, what type of proteins are they expressing. You can quantify the amount of complexes, look at their protein organization, etc. Okay, so in this second application example, I would like to move more into how you can use this technology in cancer research, and particularly for predicting drug responses. So traditionally, drug response prediction has been done with transcriptomics, proteomics, different ways of measuring either transcript or protein abundance. But here you will be able to use this spatial information to predict drug responses, giving you completely new dimensions and actually much higher efficiency. So this particular project was a collaboration with Aaron Vinney Lab at Columbia University, and they were interested in looking at AML patients and to predict response to drug with the pre-treatment acquired samples. So basically they acquired samples from either 5 responders or 6 non-responders prior to treatment. And then they followed these patients, they looked at how they responded after therapy, and then using our method, they wanted to see, can we actually predict which patients will respond and not, using this protein abundance, clustering, and co-localization as a kind of data points. So first of all, we identified the different cell types in these samples. So these were bone marrow samples with heterogeneous populations. And we noticed that the percentage of AML blasts within these samples were actually differing between the responders and the non-responders. So on average, the non-responding patients had much higher numbers of blast cells, which is maybe not so surprising. However, this information was not really enough to predict responses. You see that you have some patients with very high blast numbers that were still responders, some patients with quite low blast numbers that were still non-responders. So it was really not enough to predict the individual patient response. So instead, we used this machine learning approach where we fed the pipeline with both abundance, clustering, and colocalization data. And then in a non-supervised way, so basically this algorithm takes all of the information and it tries to separate the patients into responders and non-responders. And doing this, we could achieve actually a very high efficiency and accuracy in the response prediction. And when we later asked the algorithm, what features did you actually use to predict this response, we could see that the algorithm was using both the protein abundance, but also information about protein clustering and colocalization, really providing much higher accuracy for this prediction than if you would only use the protein abundance. So going into one more kind of cancer or hematology application, this was a collaboration with the Richard Rosenqvist Brandell lab at Karolinska Institute in Sweden. And they were interested in knowing, can we use this technology to better understand the development of drug resistance or acquire information about the mechanisms behind drug resistance in CLL? So what they did is that they took some CLL cells in vitro, and then they treated them with 3 different drugs that are lethal to CLL cells. But they treated them at a subtherapeutic or sublethal dose for several weeks, and this induced resistance in the cells. So after these weeks of treatments, they were interested in comparing the resistant cells to the non-treated cells and seeing what happens to these cells' protein interactome and what does this tell about the resistance development. So here I'm showing you differential colocalization scores. So basically, if you have red scores here, it means that these proteins are all having higher colocalization in the resistant cells as compared to the non-treated cells. And if you have blue values, so then it means that you have lower colocalizations of these proteins in the resistant cells as compared to non-resistant cells. So just looking at these 3 different drugs, you can see some patterns that are really shared between all the drugs. So for example, we have a reduced colocalization between these different tetraspanins and CD19, which is a very important functional protein for B cells. So all of these drugs are actually inducing reduced colocalization of these proteins. So this seems to be really a common mechanism for the resistance development. On the other hand, you can see some drug-specific responses. So for example, you see these proteins, different adhesion and lipid raft-associated proteins that are increasing in colocalization in the resistant cells, but only in response to the BTK inhibitors. So both of the 2 BTK inhibitors, whereas the BCL2 inhibitor did not really show any changes in these proteins. Also showing you how you can acquire really mechanisms that are specific towards specific drugs. As a second part of this project, this group were interested in profiling the cells from patient CLL samples. So as a first step of this project, we just made sure that they could really efficiently detect their CLL population. So we performed some high-dimensional cell clustering based on a protein abundance to really detect all of these different cell types. We also correlated these results to flow cytometry. So similar to what I showed you in the introduction, we could retrieve very similar results for the cell identification, either using our method or flow cytometry. Here you can see profiling of 3 different donors and a lot of different cell types, showing you very high correlation. Of course, since this is a 155-plex panel, it allows you to identify much more cell subsets than you can with the typical flow panel. So in normal resting PBMC, we usually identify around 35 traditional cell populations. So once we had made sure that we can identify the cell population of interest, we went on and we looked at, okay, so in patients that are relapsing in CLL disease after treatment, is there any changes in the protein organization? And can we use this data to identify new biomarkers of disease relapse? So basically they provided samples of the patients that were acquired either before treatment, during the response, so when the patients were actually in remission, and as well when the patient were relapsing. So we compared the relapsing CLL cells to the cells in remission, and we could see quite a few proteins that were different, both in terms of their protein abundance, but also in terms of their organization. So one protein that we found really interesting was CD352 or SLAMF6. So this is a protein that is known to regulate the survival of B cells, and we could see that this protein was showing a very clustered phenotype only in the relapsing cells and not really in the cells in remission. So this could be a potential new drug target trying to inhibit this increased clustering. So the last application example I would like to show you is looking at lymphoma profiling. So in this particular project, there was a collaboration with Illumina. We were interested in looking at how does Burkitt lymphoma cells differentiate or how is their protein organization different from healthy B cells? And can we use this information to actually identify bispecific targets for drug therapy? So in the first part of the project, we were very much interested in understanding mechanistically CD40. So CD40 is a potential therapeutic target in Burkitt's lymphoma. So it is expressed in cancer cells, but also in healthy B cells. And it's quite interesting because if you target CD40 with a therapeutic antibody, you can get very differential responses in different cells. So in healthy B cells, you typically induce proliferation by this targeting. On the other hand, in some cancer, you can actually induce apoptosis of the cells when you target CD40. And it's not really well understood why you get these differential responses. And this is part of the reason why it's been difficult to get CD40 treatment to actually work in clinic. So what we did is that we compared healthy B cells to 3 different Burkitt lymphoma cell lines. First of all, just looking at CD40 expression, and it was very high in all of the different cell types, particularly high in Raji Burkitt lymphoma cells, but the Ramos, the Dowdy, and the healthy B cells all had very similar expression levels. So this did not really explain the differential responses. So then we looked at how is the proteins organized on these cells. And just looking at this from a distance, I think you can appreciate that every single cell type actually has a unique profile. So the complete organization is very unique for these cells. There are some features that are shared. For example, you see this multi-protein domain is present in all of the cells. And we also noticed this multi-protein domain that was present in the healthy B cells, but completely absent in all 3 of the cancer cell lines. So again, providing you kind of an example of a biomarker that you can use based on protein organization. We then focused on this multi-protein domain, and we noticed that CD40 was part of this domain. So in healthy B cells, CD40 was co-localizing with this domain, being rich in tetraspanins and in another therapeutic target, so CD20 that I was presenting earlier. On the other hand, in all 3 tumor cells, you could see that CD40 was actually excluded from this multi-protein domain, really showing a completely different organization as compared to healthy B cells. And this could potentially explain the differential responses to CD40 binding that you have in healthy B cells as compared to the cancer cells. And in the second part of the project, we wanted to look at these cells and see, can we identify any bispecific targets? So basically, what I mean with the bispecific target here is a target consisting of two different proteins that if they are co-localizing, you could target them with an antibody. And if they are not really co-localizing, you wouldn't really get efficient binding of your antibody. And this could enable you to target proteins that are expressed both on healthy cells and cancer cells, as long as they are co-localizing only on the cancer cells. So we identified an example of two of these targets, so CD38 and CD37. They were expressed both in the healthy cells and in the cancer cells. But in the healthy cells, these proteins were not really showing any co-localization. In the cancer cells, on the other hand, you could see positive co-localization between these proteins in actually all of the three tumor cells. So showing you an example of one of these kind of cancer-specific co-localization or cancer-specific interactions that could be targeted. So now I've just showed you some example within the field of immunotherapy and hematology, but we do have customers doing all kinds of research in basic immunology, in autoimmune disease, really understanding drug mechanism of action. So as I mentioned before, it's really any setting where you have immune cells, you can use this new dimension to really get completely novel insights. And just to summarize, what we provide is then this sample prep kit. So it allows you to analyze 155 proteins and looking at protein interactomics, and it's completely sequencing based. One of these kits makes it possible to look at 8 different samples, so it's 8 reactions per kit, and you can analyze up to 1,000 cells per sample. So with that, I would like to finish this introduction to the technology, and I'm really happy to take any questions that you might have.
Okay, thank you so much, Sandra. I really appreciate it. Now we are move on to a Q&A session. From this session, is Dr. Luis Reinanke also join us. Thank you so much for joining, Luis.
Thank you.
Okay, so let me check the chat box and the Q&A. There's one question. It's in the example identifying tumor patches, you were detecting the proportion of doublets. What method do you use to calculate the doublet triplet rates? Can you answer this?
Yeah, sure. Maybe I can actually get back to the presentation. It's a bit easier.
Okay, please.
So in terms of the doublet detection, this is based on, you can use different methods to doing this. You could, for example, use kind of gating, looking at which, if a conjugate of cell is expressing both T-specific and B-specific markers. So here, for example, we're plotting CD20 against CD2. So CD20 expressed on the B cells, CD2 expressed on the T cells. You can see that you have some cells here that are expressing both. So this is basically cells that are positive both for T cell and B cell markers. This could also be cells that have gone through a lot of trogocytosis. So in order for you to separate the cells that are true conjugates compared to those that are going through a lot of trogocytosis, you can visualize the cells and you can also count the number of patches. So we have automatic methods for doing this. So this allows you to see, okay, is it a cell that has just gone through a lot of trogocytosis? Then it will have several tumor patches. On the other hand, if it's a true conjugate, you will only have one tumor patch, but it will contain a lot of molecules. You can count the number of molecules and make sure that it's a big enough number for it to really be a true cell. So that's how you can differentiate those two instances.
Okay, thank you so much. And if the participants have an additional question, you can use raise hand function to ask directly. Okay.
Oh, sorry, I missed the question. Can you repeat it?
No. This is not a question. This is just an explanation about the.
Okay.
raise hand function. Yeah. Okay, so actually, we received some question in advance, and let me share my screen, my slide. Yeah, so actually, received a question, and the first question is, are there the mouse panel available now? Or can you answer this question?
So the panel or the product that I presented today, it's targeting human proteins, so it's only possible to analyze human samples, and we currently do not have a mouse panel available. So this method is it's really made for human cell analysis.
So do you have plan to develop for the mouse sample?
Yeah, so technically there is no limitation to that. We would just need to incorporate mouse antibodies. It is part of, we have got this request a lot, but it's not in the pipeline for now. So it's focusing on human samples for now.
Okay. Okay, the second question is, do you have customer service to make a custom antibody?
So typically we do recommend customers to use this 155-plex panel. It is very broad, so we have got the experience that in most cases customers are happy with this panel, and it really allows you to discover quite a lot about your cells. This panel is available online. You can see exactly which proteins are part of this panel, that is kind of the product that we provide. Of course, we continuously have developments. Our previous product, it contained 80 markers. This new product now that we have is 155-plex, so we are continuously developing the panel. So we're also really happy to get input from our customers in terms of targets that they are missing. So feel free to reach out to me if you have particular proteins that you're very interested in assaying, and we will take that information for future development.
Okay, thank you so much. And the third one is, do you have some use cases using solid tumor samples? Yeah, so you explained only a PBMC case and the hematology case, right? So yeah.
So this method is made for cells in solution. So the cells needs to be in solution for it to work. However, you could use kind of solid samples. So for example, we have done a application example with spheroids. So where we were co-culturing breast cancer spheroids with immune cells, but then at the end of the protocol, we dissociated these samples. And after dissociation, you can analyze your immune cells. We have also had some customers doing this on melanoma samples where they have dissociated the tissue, looking at tumor infiltrating immune cells. So as long as you dissociate the cells, it is possible to do it, but they need to be in solution.
Okay, thank you. The first one is about the sequence depth for the analysis. Yeah.
So in order to really build this complete network of the cell, you need to perform quite high or in-depth sequencing to really capture all of the protein links. We are identifying around 40,000 molecules on the cell surface, and all of the links between them needs to be captured. So we typically recommend around 250,000 reads per cell. That is usually appropriate to get a really good coverage of the entire cell surface.
Okay, the next question is regarding assay principle.
Sorry to interrupt you. I just wanted to mention that regarding the sequencing.
Oh, we cannot hear you now, Liz.
Can you hear me?
Now I can hear you.
Yeah.
Sorry. I just wanted to mention regarding the sequencing that it is possible now to also sequence with MGI. So most customers have been sequencing with Illumina, but you can also sequence with MGI. We recently started with that. As long as they are using some sort of conversion kit, which I think is quite common, that also provides really nice results.
Great. So you have already released the additional kit for the MGI.
We don't provide the conversion kit ourselves. What the company that we have been working with, they have a conversion kit at their premises. So we give them the libraries, and then they use the conversion kit, and then you can sequence on MGI. So it's been working really well.
Okay, nice. Great. Next question is about as a principle on how to prevent a self-link between linker sites within the same rolling cycle probe.
So this is a very technical question.
Yeah, technical question.
I don't have any slides. Sometimes it can be a bit easier to explain this with illustrations. We do have that in the preprint, so that also explains this a little bit more in detail. Just in short, basically how we do is that we have two different types of DNA sequences, and you can never link one type with the same type. So you need to link the two different types, and this makes it then not possible for one rolling circle product to link with itself because it is of the same type. So that's very short explanation, but I would really recommend to check the preprint to really understand how this works.
Okay. Next two questions about the data analysis and are the analysts available, and is it possible to analyze the data in Sura or other popular single-cell analysis?
So the pipeline, we have developed a pipeline that basically allows you to go from your sequencing reads and then to regenerate these surface maps on the cells. So it is called Pixelator. It's a completely open source software that you can just download. So once you have done this step and you have converted the reads to kind of single cell data, after this step, you can use traditional single-cell analysis pipelines, so Seurat or ScanPy, and the analysis is very similar to the type of analysis you would do for single-cell RNA sequencing, for example. So you can usually use all of the same visualization tools and things like that. We have also made tutorials for data analysis that we have available. So we really try to be very assisting in the data analysis pipelines. This is a completely new type of data. It is, of course, a lot to dig into, but the way that the data is shaped is very similar to other single-cell data types.
Okay. Yeah, and the last one, the related question, the data integration with single-cell analysis data.
So we have some customers who have done that for the same samples. Of course, you cannot do it for the same cells. You cannot run single-cell RNA-seq on the exact same cell that you analyze here, but you can split up your sample. So if you have a sample, you could divide it into some cells that you use for single-cell RNA-seq and some that you use for the proximity network assay. And then you can compare the results and integrate the results with that.
Okay. Okay, thank you so much. And let me check the chat box. It's no question now. Yeah, okay. Okay, any other questions? Okay, let me allow to greet in Japanese lastly to the audience. Yeah.
Yeah.
Okay. 本日大変お忙しい中ですね、ご参加いただきまして誠にありがとうございました。ちょっと分かりにくい部分とかですね、あったかと思いますけれども、これレコードしてですね、また公開をさせていただきますので、また弊社の方からですね、メールにてお知らせをさせていただければと思います。ご質問等ございましたら、弊社バイオストリームの方にですね、どんなことでも構いませんので、お問い合わせをいただければと思います。本日は大変お忙しい中、本当にご参加いただきまして誠にありがとうございました。じゃあ、今日はこれで失礼させていただきます。ありがとうございました。Thank you so much for everyone. Yeah. Have a good day.
Thank you very much.
Thank you.
Thank you. Thank you. Bye-bye.
Bye.
Thank you, Naya. Bye-bye.